Okay, this is going to be a slightly heavy video, so buckle in. For this particular video, we are going to be talking about B lymphocytes and their immune response towards antigens or any foreign materials in your body. Before we talk about that, just a little bit of brief revision. We have to talk about the introduction to B lymphocytes. They are formed in the bone marrow from blood stem cells, and they become something known as immature B lymphocytes. And they will undergo the maturation process in the bone marrow as well. That is why they are known as B lymphocytes. We talked about this in the previous video when we were comparing B and T lymphocytes. After becoming mature B lymphocytes, obviously the size becomes bigger, as you can see the comparison left and right. A very important thing I want you to know here is that these B lymphocytes are all genetically identical to each other. Genetically identical meaning to say, if we compare the chromosomes in the nucleus, they will have the same number of chromosomes and the base sequences in each chromosomes are exactly the same. And that is why in some situations, we refer to all these mature B lymphocytes as clones because, they are identical to each other. However, as much as they are referred to as clones, what will happen is during the process of maturation, they will also develop receptors on their cell surface membrane. Now, here's the weird thing. Even though the clones are all genetically identical, they will produce different types of receptors. As you can see in that diagram, compare the shape of the receptors of those 4 mature B lymphocytes. Are they all exactly the same? No. Each B lymphocyte only has receptors complementary to one type of antigen. So this B lymphocyte, all the receptors on its cell is exactly the same, which is complementary to one type of antigen, and it's the same for the other 3 as well. Now, some students will say, oh, does my body only produce 4 mature B lymphocytes? No, your body actually produces millions of different types of B lymphocytes. I'm just condensing it into just 4 types because, we can't draw all 1 million. So, what do these B lymphocytes do anyway? The thing that you have to understand is, in the beginning, when you're born, you have very little concentration of each B lymphocytes. And they are usually dispersed all around your body. Some are inside your blood, some are in your lymphatic system, some are in a place called the lymph nodes. You don't need to know that in detail. But, the point of the matter is, you start off with very little B lymphocytes. That's basically it. Let's imagine for a second that I'm representing, okay, you look at the human body here. Now I'm putting 4 dots there. Those 4 dots imply that you have very few B lymphocytes at the very beginning. That's what it means. It doesn't mean that the B lymphocytes are in your abdominal area. No, it doesn't. It is just a symbol, or it is just a representation to just show you that in the beginning, before any immune response, you have very few B lymphocytes. That's basically it. Okay, you don't need to. It's good to understand this part first. All right, but the B lymphocytes are actually circulating all around your body, by the way. But I'm just, let's just imagine that they are there at the center area of your body. Now, let's talk about the immune response of the B lymphocytes. You see, B lymphocytes will only produce an immune response when antigens are present. So, and where are the antigens? Antigens are usually located on pathogens or harmful microorganisms. So let's start. Now, when the pathogens first infect your body, let's say this is a type of pathogen like a virus, okay? So the virus just goes into your body and it infects your throat, all right? Represented, as I've highlighted, that green color area at your neck. Okay, so when it infects your throat, are there any white blood cells nearby? based on this diagram? No, there are no white blood cells nearby because, as you can see, the pathogens are over there, and the mature B lymphocytes, because their concentration is so low, they are quite far away. So the pathogens are like, there are no white blood cells over here. I can cause havoc in the body. So what do the pathogens start to do? Well, the pathogens will then, start to infect your body cells, and as they infect your body cells, they are stealing the energy from your body cells, they are stealing the nutrients, and they are also reproducing or replicating, and the number of pathogens increase in your body, as I've represented in those green dots. So you can see those green dots, they are starting to increase and they are starting to spread in your body. Now, even as they spread inside your body over here, have they met the lymphocytes? Not yet. They're still a little bit far away. In this case, as the pathogens increase in number, the person falls ill. And if the person falls ill, they will show symptoms. For example, if this were COVID, you will start having fever, you will start to cough, difficulty breathing, those kind of things may happen. If this were tuberculosis, you would start to cough as well, you will start to have chest pain, night sweats. Or if this were cholera, yes, I know cholera infects the small intestine, I get it. But imagine if this were happening, the person may get diarrhea. So this is the body developing symptoms towards the infection. Now, during this period of time, of course, the person's body is being damaged, and eventually what happens is, as you can see here, the number of pathogens increase further, and a few days later from the first day of infection, a few days later, the pathogens and B lymphocytes eventually meet. They don't meet immediately because, as I've said to you, you had very little concentrations of the B lymphocytes at the very beginning. Now, here's where it becomes very important. So how does the B lymphocyte respond to the pathogens? The first vital thing to mention here is, not all B lymphocytes can respond to the pathogen. Why not? Well, look here, it's very obvious. Look at this B lymphocyte. Can they respond to the pathogen? No, because the receptor is not complementary to the antigen. Second one, again, not complementary to the antigen. Third one, again, it almost looks the same. Okay. Oh my God, my B lymphocyte got squished. It's not complementary to the antigen. But that fourth B lymphocyte, remember, it was inactive, but the moment it binds to the antigen, the moment the receptor binds to the antigen over here, that is what stimulates that particular B lymphocyte. So out of all the clones, only one specific B lymphocyte was stimulated. And that process is called clonal selection. Why is it called clonal selection? Clonal meaning to say the clones, okay? And out of all the clones in that group there, only one of them or one type was selected to respond to the pathogen. And so in this case, the B lymphocyte with receptors complementary to the antigen will be stimulated. And when they are stimulated, they don't immediately kill the pathogen. You see, the B lymphocyte does something pretty fantastic. What it does is it starts to undergo a process known as clonal expansion. So only that B lymphocyte that was stimulated. So expansion means, you might be thinking, expansion, it becomes bigger. No, in this case, expansion means it will undergo mitosis repeatedly. And mitosis is the process of nuclear division and cell division to produce genetically identical cells. We saw this in chapter 5. And when they continue dividing, they will form a lot of cells, but those cells are divided into two groups. And the two groups are memory B lymphocytes, and they will also form these important cells known as plasma cells. Now, what are the functions of these plasma cells? So these plasma cells, which I've represented, can you see those those green colored dots over there, okay, at the bottom, and I'm representing it, I'm highlighting a pink color area, it doesn't mean that the plasma cells are located on that part of the body. I'm just showing you that your body is producing some plasma cells, right? So those plasma cells, what they will do is they will then secrete antibodies. Please do not just say they produce antibodies. Production just means the antibodies are still within the cells, but they will produce and secrete or release those antibodies. We will be talking about what antibodies are in another video, but the point of the matter is these antibodies are proteins and they are complementary to the antigens. As the antibodies bind to the antigens on the pathogen, right, as you can see here, they are attacking the pathogen. Look at what happens to the number of pathogens in the body. The number of pathogens in the body starts to decrease. Now you might be thinking, oh, how exactly do the antibodies destroy the pathogens? We will be talking about that in another video. The point that I'm just trying to make over here is when your B lymphocytes produce those plasma cells will secrete antibodies complementary or matching to the antigens. And as long as they bind to the antigens on the pathogen, those pathogens will be destroyed. This is known as the primary immune response. Why is it called the primary immune response? Because it's your B lymphocytes' first time responding to that particular type of antigen. Okay, it has never responded to that antigen before in your entire life, so this is the first time you're responding to it. And the primary immune response, if you notice, it sucks because you fell sick, because the response did not happen immediately. The pathogens had time to enter your body, infect your body, and cause damage to your body. Your body only responded to the pathogens a few days later. Okay, so the primary immune response did save your body, but there was some collateral damage. The collateral damage being some of your body cells or some of your tissues that got killed in the process. Like for example, you had a sore throat, or you had some lung infection, or even diarrhea. That's not cool at all. However, here is where it becomes pretty interesting. The plasma cells only survive for a few days. Okay, the reason why the plasma cells only survive for a few days is because their function is just to secrete antibodies, and they are secreting a lot of antibodies, might I add. We would approximate and say like they are secreting about 1,000 antibody molecules per second. And to secrete those antibody molecules, to produce and secrete those antibody molecules, your body needs a lot of energy. That is why I always tell, this is not a medical advice, by the way. I'm not a doctor, but one of the most important things to do when you're sick is to make sure that your body has enough energy and the proper nutrients. So you should be eating a food with a lot of carbohydrates and proteins to nourish the immune system so that we can efficiently produce the antibodies. Now, so they will die off eventually, and that's fine because the infection has been eradicated, the pathogen has been eradicated, so the plasma cells do not have any more functions anyway. But the interesting thing here is the memory B lymphocytes, as you can see, I'm just circling it over there, they remain in the body for a long time. Now, and look at the number of memory B lymphocytes, there are more. And those memory B lymphocytes are now they are now circulating in every part of your body, in your blood circulatory system and your lymphatic system. And I'm representing it here in our body as those orange dots. That means the point here is, in the beginning, you had very little B lymphocytes, but now you have more of those specific B lymphocytes in your body. Not all, but just those specific B lymphocytes with that receptor shape. Now, why is this such an important thing? The reason is because when the pathogen infects the body for the second time, okay, see, the first time it infected your body, the pathogen was very far away from the memory B lymphocytes, so they didn't know what was going on. But because now you have so many memory B lymphocytes, so what is the function of these memory B lymphocytes? A lot of times my students will love to say, well, they remember the past infection. And yes, I know why a lot of students say that, because the word memory is there. I understand that, but that's not how the immune system works. There is no actual remembering. Right. And personally, on a personal note, I hate that word memory B lymphocyte. It implies that your immune system remembers when it doesn't actually. You see, the first time the pathogen entered your body, you had very little B lymphocytes. So your response to the pathogen was not immediate. It took a few days for it to happen, right? But the second time it happened, see, you have so many memory B lymphocytes circulating in your body right now that the moment the pathogen comes into your body, what happens? It's like, oh, shoot, there are so many B lymphocytes in your body, and each of those B lymphocytes will immediately latch on to the pathogen. In fact, it doesn't take a few days, it might just take a few hours for it to happen. When the pathogens infect your body for the second time, the same type of pathogens, by the way, your secondary immune response is much faster because you are responding to the same type of antigen for the second time. And the reason why it's faster, it's due to the memory cells. So if a question asks you, what is the function of memory cells? Do not say that they remember the infection. You have to say that it produces a faster immune response in the future if the same type of pathogen infects the body. So the pathogen doesn't have time to cause an infection and, increase its number inside your body. If you saw in the first immune response, how many B lymphocytes responded to the pathogen? Only one. But now you have 3 memory B lymphocytes responding, and all 3 of them will undergo clonal expansion. So there is clonal selection of the memory B lymphocytes and clonal expansion. And if all 3 of them undergo clonal expansion, you will produce even more memory B lymphocytes, and you will also produce even more plasma cells, much more plasma cells. And when you produce much more plasma cells, guess what? The function of the plasma cell was to secrete antibodies. The first time you got a little bit of plasma cells, it released a little bit of antibodies. But the second time, because you have more plasma cells, it releases way more antibodies to defend your body against the infection, as you can see here. So the pathogen has like you see, there's only one pathogen over there, but your body released this is an overkill. Like your body produced way too much antibodies to kill the pathogen. Now, is this a good or bad thing? For our level, we will assume that this is a good thing. There can be some detrimental side effects if your body produces too much antibodies, but we will not be going into that. The point here is, at least on this level, we should just assume that the more antibodies you produce, the better. So, in this case, the person did not fall ill at all. And why did the person not fall ill at all? Because the pathogen the pathogen went into the body, but the pathogen had no time to cause any damage on the body whatsoever. So this is good, and this is known as the secondary immune response. So in the primary immune response over here, let's just focus on that B lymphocyte. Look, you had very little B lymphocyte to begin with. Okay, I'm just representing it as one B lymphocyte. So when the pathogen enters the body for the first time, do they meet the B lymphocyte immediately? No, they don't, because there were very little B lymphocytes to begin with. So the pathogens had enough time to replicate or reproduce. And as they replicate and reproduce, they will start to cause damage in your body, symptoms. So you'll start to have symptoms of the infection. But a few days later, what happens? That specific B lymphocyte will meet with the pathogen, and something known as clonal selection takes place. Clonal selection just means that the B lymphocyte with receptors complementary to the antigens are stimulated. And when clonal selection happens, it is followed by clonal expansion, where that B lymphocyte undergoes mitosis to produce two types of cells, memory B lymphocytes and plasma cells. And those plasma cells will secrete antibodies. And as your body secretes antibodies, look at what happens to the number of pathogens. The number of pathogens will start to decrease. Why it decreases? Again, we will talk about that in a different video. Don't worry. But the important thing to understand here is the person was sick in the beginning due to the high number of pathogens, but after a few days, the person recovers. And as the person recovers, the person has more memory cells in the body. And those memory cells I'm representing as those dots. So let's just say a few months later, the same type of pathogen with those same specific antigens enter the body again. Will the body take a few days to respond to the pathogen? No, because as you can see here, there are a lot of memory B lymphocytes circulating inside the body. So the memory B lymphocytes respond almost immediately or much faster. A faster immune response within a few hours. You don't have to say that it responds within a few hours. You just have to say that the memory B lymphocytes produce a faster response. That's good enough. Okay, and in this case over here, because more memory B lymphocytes respond immediately, your body will produce even more memory B lymphocytes, okay, and the body also produces more plasma cells. And because there are more plasma cells, there is a secretion of a higher concentration of antibodies. And in that kind of situation, the pathogen is immediately eradicated. And the person did not fall sick at all, okay? So here's the interesting thing. As your body gets infected by the same type of pathogen, more memory B lymphocytes are produced, even more, until it reaches a cap. But the point is, your body gets better at responding to that pathogen. In the words of Friedrich Nietzsche, who was famously quoted as to say, what doesn't kill you in this case makes you stronger. Because the pathogen did not kill you the first time, our lymphocytes are now better at responding and destroying the pathogen if it tries to enter your body in the future. So the one at the top is the primary immune response, and the one at the bottom is the secondary immune response. And as you can see here, after the end of the secondary immune response, you have even more memory B lymphocytes, like way so much. So if the pathogen tries to enter your body the third time, immediately destroyed. And that pathogen is no longer a problem in your body. So the point I'm just trying to make here is, if you look at this graph here, when the pathogen enters the body for the first time, let's say January 17th, what I mean there is the 17th of January. Okay, it's a little bit confusing, I'm sorry. So on the 17th of January, for example, let's say you had a first infection. But does your body respond immediately? No, because the first time you had very few B lymphocytes. So you only responded to the pathogen on the 21st of January. And when your body produces, your body will secrete, your plasma cells will secrete some antibodies in the beginning, as you can see there. Wherever I'm circling, that responds to that part of the graph. And after the antibodies bind to the pathogens are then broken down by the phagocytes and also your liver. So that's good, that's fine. And after, they will decrease and your body has eradicated the pathogen. That's good. Now, a few months later, let's say the 21st of June, your body had a second infection of the same type of pathogen, right? But in this case, did it take a few days? Did it take 4 days to respond to the pathogen? No, it didn't, because you had memory cells, and the memory cells respond almost immediately. Look at the concentration of antibodies released in this case. It doesn't release very little antibodies, it releases a lot of antibodies immediately, because you had more memory cells, and more memory cells undergo clonal expansion to produce even more memory B lymphocytes and even more plasma cells. And when you have a higher concentration of plasma cells, obviously there are more antibodies. That is why the graph looks like that. But that is why the antibody concentration in the blood is more during the secondary immune response. So during the primary immune response, yes, there was a delay and you did not produce a lot of antibodies, okay? But in the secondary immune response, it was much faster and a lot of antibodies are secreted because you had memory cells. So during the primary immune response, you might have fallen sick. You might have been coughing or you had the symptoms of the infection. But the secondary immune response, you were like, oh, was I even infected? You don't even know that you were infected at all. Like you were not aware. There is a war that is constantly going on inside your body that you are not even aware of. The reason is because the war immediately ends due to the presence of your memory B cells. The memory B cells produce a faster response, and you were not even aware that you had pathogens in your body. I always tell this to my students. The reason why you're not sick is not because pathogens are not in your body, but because the immune system is constantly functioning at all times. This is what the immune response of the B lymphocytes are all about. Now, I know, yes, it's a 30-minute video. I get it, but I didn't want to break down this video. I just wanted to condense it together because B lymphocytes are the most complicated thing that we have to talk about in this chapter.